Warm welcome to IRIS Animal Sciences. Yeah, but animal sciences doesn't mean a very stricto sensu invertebrates, chordates in that way. Animal sciences are integrated with several branches. Like Mr. Shrey was just talking about various branches. He was talking about molecular biology can be related to animal science. Your environmental sciences can be related with animal sciences. Uh, Biomaths can be related with animal sciences. There are so many subjects in that which you happen to see those branches can be directly or indirectly related with animal sciences. Basically, once you start doing the project, if there is more weightage towards animals per se, then it will be classified in the animal sciences. And if there is more weightage on the other subject, based on your own suggestions, we may try to adopt it into some other subject. So animal sciences is a very open, huge, wide platform on which you can understand and do your work. Basically, I wanted to share with you as to research how and why. Why should I do research? Why should I do it? Children, please understand that research is a creation of new knowledge. That is innovation. But you can also use existing knowledge in a new and creative way, right? Please remember that. So it's sometimes extremely an innovation, or sometimes it can be generating new concepts, methodologies, understandings. Only thing is, as long as we are innovating, we are growing. So you have a very important role to play in the IRIS program. Please concentrate on how to think and not on what to think. So start thinking today. Start thinking. Think differently. Think differently. Wonderfully well. A simple question for why research. I'll just tell you a simple example. Why should we do research? Why should we think in an alternative way? The general question which I put forth for children, when I ask you people who eats more, you normally tell that the elephant eats more than a dog. What I want you people to understand is when I start looking at the weight of an elephant, the weight of a dog, how much that grass does the elephant eat per day, how much meat a dog can eat maximum. And then if you understand science per se, if 5,000 kg elephant eats 50 kg, 1 kg body weight of elephant eats how much? It eats only 10 grams. Whereas that of a dog eats 50 grams. So who is eating more? Why? That is research. Why should a dog eat more than an elephant? It's because elephant is at a trophic level, which eats the grass, which fixes the energy, whereas a dog eats the meat of an animal, which eats the grass, which fixes the energy. These are simple examples. So keep questioning. Keep questioning, children, and keep questioning, and prepare a mile chart now itself. That I am going to do something, and these are the methods I'm going to follow. Yes, the moment you start, you will get a confusion. Oh my God, how am I going to do this? If you are confused, I congratulate you, because confusion is the beginning of clarification. So be happy. Start planning your research first. Put a plan. Doesn't work out, don't bother at all. In English language, there are 25 more alphabets. No, put plan B, plan C, plan D, plan E. So what? You have time. Start working on it now. Challenge yourself. That's the most important thing. So first step, what you will do is in animal sciences, or whoever children who are listening to me today, in whichever branch you would like to work on, identify and develop your topic. What am I interested to work on? How can I develop my topic? Right? For that, what should I do? I must find background information. Where can I find background information? I must do proper literature survey. Children's very important literature survey, right? You have to make literature survey. You can have internet sources as well, and original research papers if possible. That is very important, because sometimes we have the habit of referring back references, and they may not be reliable sometimes. So original research papers. Okay? Then I select a problem. Once I select a problem based on literature survey, based on availability of resources, based on books, based on inquiries from people, or whatever is required as a social requirement, and also through Google. You can find sources, what all things you can do from any of the sources libraries. And then you can define your research into different branches. It can be exploratory research, which structures and identifies new problems. You are identifying a new problem. Now, for example, COVID came in and COVID has almost gone, right? Then you can find out what is the impact of that on some organic organisms, how animals are reacting to it. Are there any sort of differences, possibilities, anything you can work on? That is exploratory research. Or you can do constructive research, providing solutions to problems. It was an already existing problem. You are trying to provide a solution to a problem. Or you can do empirical research. Test the feasibility of a solution using evidences. You define yourself in whatever way you would like to work on. But don't expect miracles. This doesn't happen in science, okay? You can't mix 3 green apples with 1 red apple and produce an orange out of it. It's not possible. Step number 3, very, very important. What are the materials I would be using? And what is the methodology I'm going to follow in my research on animal sciences? If I'm going to work on certain aspects, how am I going to work on? How am I going to do it? How am I going to use it? Right? And during material methods, you must design your experiments. And children always remember, before you design itself, have an idea about the statistics you are going to apply into it. Many children, why many children? Even I, when I was doing my research, we don't try to work on statistics because we were allergic to mathematics. That's why we took animal sciences, right? Fine. But we, at the last moment, go to a statistician and they are not satisfied with the data which we have generated. So while designing the experiments itself, try to have some idea about statistics, whether you will be going in for significance or correlations, whatever values you would like to apply. Sample size is very important. Remember that. Depending upon the type of experiment, how many number of samples and repetitions I have to do. The replicate should be there. Control should be done. Supposing I want to study about giving a feed to a bird or a fish and how much it is growing, then I must also have controls where I give the regular feed and where I don't give any feed. So one is a negative control, one is a positive control. So while you plan your experiments itself, please see to it in any of the subjects you do. When you plan an experiment, there should be proper controls included. Okay, Raja? Let's start example. For example, I am taking for example, vermicompost. Now, vermicompost is animal derived, but I'm going to give it to a plant. Now, when I'm giving it to a plant, I grow a plant. Yes, this is how I'm growing. I'm going to grow a plant. Fair enough. Fair enough. But I'm going to give something. Now, when I'm going to plant 2 types of pots, am I comparing? Are they equal? No, they are not equal. Why they are not equal? Because the quantity of soil in one experiment is more, in one is less. If I'm doing an experiment where I'm having the title as the volume of soil on the growth of the plant, then this experiment is right. If I'm going to measure the plant growth based on the soil use, then this experiment is wrong. Is that clear, children? I should be very clear as to why I'm using the experiment, for what purpose I'm going to do the experiment. In the same way, when I use soil and I'm going to study the plant as well, if I'm going to study the plants growing on different types of soils, then this experiment is right. If I'm using the growth of plant irrespective of the soil, then this experiment is wrong, because one is brown soil, one is red soil. Is that clear, children? We should be very clear when we set up our experiments. Similarly, I'm going to give vermicompost to both the soils. Both are perfect. Both the experiments are very clear. But for one, I'm able to dump more compost. For the other, I'm giving less compost. If I'm going to do an experiment based on the volume of compost used, then both the experiments are, this experiment is right. But if I'm just putting compost and seeing the growth of the plants, then this experiment is wrong. So I should be very, very clear what is my objective and what type of experiment I'm doing. Especially because you children, you are interested in animal sciences. Whenever you're going to do an experiment using live animals, you should be very clear, children, that the laboratory should be recognized by CPCSCA. Now, what is this CPCSCA? It is a committee for the purpose of control and supervision for experiments on animals. That lab should be certified. Now, your school lab may not be certified, right? So in that case, you must get associated with a lab which is certified, and they must give you a letter that you have worked in the laboratory. Don't rush in the last moment. You should be very clear when you're doing all these experiments. Very clear. Even in fishes, for example, I'm going to do something related to fishes. Just for the fun of it, oxygen consumption based on different stress, I'm going to study. If I'm going to use two distinct species and study, then this is correct. If I'm just putting a fish for the sake of using a fish, then this experiment is wrong because they are two different species. You should be very clear which species you are using. And you must have the species identified by a known zoologist who should give a certificate that you have used that particular species. Because with reference to every species, things may vary. Similarly, I can't use fishes at random. If I'm going to use the species, then the size also matters. Because at different growth rates, different growth stages, the consumption of oxygen or metabolism, everything varies, children. So when I'm going to do an experiment, a single way of oxygen consumption in this particular species, then my size should be uniform by weight. Now, in case I'm doing different stages are consuming different levels of oxygen, then this experiment is right. So how you use your experiment, what is the methodology you use, is all very, 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 very important. Very, very important, okay? Minimum, we expect 3 values. But because it is life sciences, Raja, normally, animal may not cooperate, plant may not cooperate. So it is better that you do several replicates and choose the best of them, what you want. That's the best way to do it. Is that clear? Right? Good. Results is very important. Be genuine and honest in your results. Children, we have been working on this for so many years, and when you come and present your data, we can easily find out if your data is genuine or not. So please be very honest in your data collection, in your calculations, applying statistics, graphic representations. Many children get confused while you give your graphic representations. You confuse between a line diagram and a bar diagram. You see, a line diagram is for a continuous process if I'm observing. If they are independent observations, then I'll have to use a bar diagram, which is called as a histogram. But if you're taking the whole muscles and studying various parameters, for example, carbohydrate, protein, lipids, etc., you can also use a pie diagram. Okay? You should be very, very clear as to which diagram you're going to use. Some simple sort of projects we can do. Well, pre-COVID, post-COVID comparison of breeding season of native birds and what is its impact on them? What happened to them? Can be a very interesting sort of it. Suppose, diabetic rats, once we start treating with the various plant extracts, for example, many people try to work out on what is happening, whether the cure. But if you can work on the defense system which is operating on it, it can be something unique for you. I'm just giving this as general inputs. You need not work on this, please. Right? Now, many of the insect pests are being controlled, and the whole world is fed up with chemical pesticides because they are very harmful. Previously, we used to call them as IPM, Integrated Pest Management. Now it is called as EPM, Ecological Pest Management. So what arthropod predators, the adult, different type of bigger insects or whatever predators, which can do a very efficient pest control and save a lot of grains, especially children. On the field, several things happen, but you should know that many of our grains after harvest are put under storage, right? Storage pest management is today becoming a big menace. If you can find out a beautiful solution for it, you have achieved it. Clear, Raja? Similarly, like tomatoes, you grow them. Every time there is some scar, some sort of a nematode infection comes. If you can find out, how can I isolate and identify and throw this fellow and see to it that we save tomatoes? Recently tomatoes, what price they were sold in the market? Right? Because tomatoes completely, we could not cultivate them, harvest them. Typical problem, this year's problem, take it up. Do it. Try to come out with a solution. Similarly, utilization of information communication technologies in agriculture. There is a lot of work. Genetic evaluation of performance of blood biochemistry, going into the details of blood biochemistry and comparing it with genomes and trying to identify these relationships can be a unique feature which you can work on, right? And also, like animals, like the moment the calf is born, it depends on the mother, that is the cow, for its milk. Then gradually it weans away. Weans away in the sense it moves away from taking milk into eating various things. Right? Now, how the genotype has all these sort of restrictions on it, no work is available today. You can be a pioneer. Effect of animal metabolism due to climate change. Nobody knows because every day we are seeing a lot of change. There's a lot of climate change. Children, you yourself know what's happening today throughout the country. Rain falls like anything in places where it's not expected to fall during a season. And sometimes it's so hot when it's supposed to be cold. So when all these things are happening, what is happening to animal metabolism also? Because all animals are not, they don't have a stereotype of temperature. They fluctuate the body temperature. So how they act on it, that can be a good beginning for you. Similarly, microbiological evaluation of raw and smoked animal food. Smoked animal food, you should know, children, that most of the fish which people prefer to in Western countries, the fish is smoked with the carbon soutes, right? This is a very big industry even in Mangalore and other places in India, where many products can be smoked and they give a peculiar flavor which people like. Recently, you will find a lot of smoked almonds now today available in the market. They give a peculiar taste. People love it. But you can find out what happens to the microbial evaluation when you store them, when you retain them, when you consume them. So much of opening is available for you. Similar effects of dietary synthetic amino acid levels. Many people market, there are several medicines today available. In the same way, they are feeding it to fingerlings, fish fingerlings. Fingerlings are fish kids. So what happens if you add different types of amino acid? Each amino acid has a different type of a composition and its contribution in the growth of an organism. Right? Similarly, the grass which is added for producing silage to feed it to the goats. Now, what happens to the goat's metabolism? How they are able to respond to it, and how this can be useful in stall feeding of animals. All these are virgin areas where you can work on. Quality and quantity of feed ingredients utilized in feed formulation. Because, like when you take any, supposing you buy a chocolate bar, you know what are the ingredients, carbohydrates, proteins, fat. Similarly, feed ingredients in feed formulation is prepared for animals. You can study that. Garlic is used as a feed ingredient. But what happens to the diet in the chickens? What happens to the load of the chickens? What happens to the internal anatomy, internal morphology, or various histochemical reactions that take place inside the gut? All these can be studied. Animal fed diets, they are also very important, containing graded levels of soybean hull. Soybean, most of the waste products are today converted into animal feeds. So what happens to them? Storage methods on egg quality. We buy eggs, we leave them inside the fridge. Sometimes we leave them in the fridge. We think that they are going on very well. We go to a tour, we come back after a month, we may even use the same egg. But what happens to the organoleptic properties of that egg? If it is a domestic fowl, that is what we call as a native chicken or country chicken. And what is the difference between the egg of country chicken eggs and the broiler egg chicken? All these are very important, right? And today, urban centers, there's heavy soil erosion taking place after the climate change. So what happens because of these erosion properties? How we can restore them? What are the building properties? How to retain them? Bioremediation properties. There are lots of such programs which you can work on, children. There is a huge, keep your imagination wide. I have just given you some 15 tips, just to make you understand that animal science doesn't mean just animal invertebrate or a chordate, or just do into physiology or cytology or genetics or embryology or evolution. It's not so. It encompasses the whole field of science. Is that clear? Or you can even prove yourself if you go to the international arena. Today, international arena, everybody, including the G20, which is happening now, talk about Sustainable Development Goals, SDGs. And there are 17 SDGs that have been identified internationally. You can take up something because the targets are very important. They say that by 2030, nutrition sufficient food. Can you find out a nutrition sufficient food through animal sciences? Many people are saying that insects are going to the future food. Have you worked anything on it? There is the BSF, black soldier fly larvae, are being converted into animal feed. But can they be a source for human consumption? I do not know. And by 2030, end all forms of malnutrition, which means you can do a lot of amino acid profiling of lots of food products and suggest what is good for the people. Because, fortification of food doesn't solve the problem. Fortification of food doesn't solve the problem. But if you can improve the soil by creating a nutritious food with very simple technologies, you can end all forms of malnutrition. Then you have achieved something great in this life, right? By 2030, the government wants to double agricultural productivity. It's impossible to double with chemical inputs today because it's saturated already. Genetic modifications are not welcome because they try to hamper with the natural products of biodiversity. But can you try to incorporate through the soil systems, manipulate the soil systems, and create or enhance food productivity to produce a wonderful future India? Because millets are being, now, for example, millets are being promoted. And I am sure, as a soil biologist, that there will be a time when you youngsters will be growing old, when India will be providing food for the whole world. Because we have such wonderful ecotypes. We have wonderful diversity of millets. Throw open, because this is the year of millets. Work on it, right? How best can I improve land and soil quality? How best can I maintain the genetic diversity of seeds, cultivated plants, and farmed animals? All these are open topics for you, where you will definitely make a mark, because sustainable development goals are the target of all the countries of the world today. Take a topic related to that and start working. But whatever you may do, meticulous observations are a must. No compromiser. And in research, it is either completely bad or it is foolproof. There can't be approximately foolproof, right? So be very clear, very honest. Don't leave anything to chance, children. Anything to chance. You take up all the pain, do the work, sit down with your team. Yes, in Iris, probably Shrey will be telling you more as and when you interact with them. You can participate either individually or as a team, but a team should not exceed 2 members. Is that clear? 2-member team. Please remember that. And that's very important for us. Discussion after you do the results, you go into discussion based on previous reports and what is your new finding, what is your innovation, and come to a conclusion. We should be precise and crisp. Children, today, bibliography references are very, very important. Whatever paper you refer it properly. Note down authors' names, published year, title of the paper, which journal published, which volume, which number, and from which page to which page. This is very, very important. We will also be asking you, yes, because we look forward to see you at the Irish National Fair. So children, be creative. Be creative. And don't confuse between education and intelligence. You may not be scoring marks in your school, but you will score with your intelligence in IRIS. Yes, I'm confident about it. It is not your marks which help us in IRIS, your intelligence, your innovation, your commitment. So research does not mean that you should put your head into trouble and start working seriously on it, but don't others operate your brain. And don't try to buy projects and sell it to us. Confidentially, I'm telling you, our SRC has wonderful investigating team. They will very easily locate if it is not your project. So don't try to buy a project or get somebody's project and try to project as it is your work, right? Never do it, never do it. And in this age of information, ignorance is only a choice. So don't be ignorant. Update your knowledge, children. Update your knowledge. Work as a team, as I told you. Do well. And then plagiarism is very, very bad. Don't copy somebody else's work, but just by changing some names and values, because we have several mechanisms to cross-check. And you all know that today AI and IoT platforms are all well developed, right? We just put your project into them. We know from where it has been copied. No copying, no buying, honest work will definitely gain our recognition, because we always remember that you are the best. So all the best, children. Good luck to you. We'll have a discussion before we wind up. Thank you.
Thank you so much, sir, for wonderful presentation. Uh, Students, if you have any question, you can raise hand and we will answer your queries. If you have any questions related to the subject or related to your participation
Or
in
iris in
IRIS,
general, anything.
yes,
Any
yeah,
doubts you have, you can ask children.
right. Also, we keep getting a lot of queries from students since the registrations are open for this year, and we keep getting a lot of emails and direct communication to us, to the team. So a lot of students are aware, and we have already started receiving.
What is the last date of submitting?
So it's 31st of October as of now.
1st of October. That's very good.
Yes, sir.
That's good.
So we this year we have opened the submission for 3 months, August, September, and October.
Very good.
Right, sir.
That's great.
So we
Children,
look.
you have a lot of opportunity. You have still a lot of time. Start thinking.
Right, sir.
Children, I haven't told you completely. Yes, Pragamya, before you ask, I just want to tell you that in case your project is selected and you go to ISEF in the US and your project is brilliant and you get a grand award, a minor planet will be named after you. And today, when we are expected to land on the Chandrayaan, be confident that you may be the next planet. Yes, Pragamya.
That's it. Unmute yourself, Prakamya.
Yes, sir. Good evening, sir.
Good evening.
Thank you so much for this brainstorming session and thank you for giving us the insight. Sir, actually, recently I'm working on a plant sciences project on abiotic stresses as we talk about climate change and global climate function fluctuations. Sir, but like when I started it off, it's been a month since I was reading literature and everything. But sir, ultimately, as you said, that as we start, we get confused, and that's what currently is happening with me. That when you read a lot of literature work, you get confused. You keep on, everything is interconnected, and that somehow looks like a big task to just interconnect different literature reviews, and then you ended up being messed up because it becomes so difficult to especially to understand the terminologies. I'm a student of class 11 and a science student. So I'm currently reading all those. Sometimes I understood something, but sometimes that a single word means have a different meaning in different literatures, and that is really a big confusion. And I really want to know how to deal with it and how to have a proper approach towards what I should work on.
Normally, in plant sciences, the word per se may be interpreted differently, but will not have different meanings as you really think here. Basically, what happens is many people publish with different parameters, abiotic stresses. Abiotic stress can be even temperature, pH, climate, various parameters can be abiotic stress over there. So what we normally do is, if you are able to identify or going to work with one particular stress, then you must create an environment where the other stress does not try to influence it. So you try to create a chamber where all other parameters are taken care of and it's not influencing. Only the parameter in which you are interested is influencing. For example, I'm going to work on the light stress on plant development. Light stress in the sense, in that case, you are going to modify it. It's a 24-hour illumination, LL pattern, or total dark, DD pattern, or LD pattern, light dark cycle. Light dark can be 12 hours, 12 hours, 14 hours, 10 hours, or 10 hours, 14 hours, right? So you create different chambers where the other things don't influence, and only the light influences. So you will have to slightly grow up. Don't go into too much strong literature. And if you're really going, I do not know where you live in, you can try to contact some professors in the neighborhood colleges or an institute who can give you the simple explanation of those papers which are being published, right? And you can easily come out with solutions. Don't worry about it too much. Don't take too much to your stress. You read them, prepare a sort of a flow chart, work out how you can take it forward and start working on it. Is that clear, Prakanya?
Yes, so that actually, so my father is a plant pathologist, so he is in that field. So he was my mentor who was helping me throughout.
Then
And
it should be easy
since
for you. What was the particular word which caused stress in you instead of in the plant?
so actually, I was reading about I was thinking of working over the functional marker system in A
Marker
biotech.
systems. Okay.
Yeah. So, these when I go through this, genes, genetics and all these, so there were the terminologies and everything that's confused that I ended up being messed up. So I talked to my teacher
I,
and
I, I,
also,
I will give you a clue here. You, without much confusing, start working on it. And in case it's something really innovative and genuine, and once it gets selected at Iris National Fair, our team will support and mentor you once you are selected. So you try to
thank you,
do
sir.
your best to see to it that it comes till the Iris National Fair selection. And from there, if it gets selected, our team will always be there supporting you. That's our
Okay,
dream.
sir.
Yes.
Thank you so much, sir.
Thank you.
Thank you.
Thank you
Okay,
very much.
so sir, with this, I would like to place a question here. So, being the chair of the committee and you know, part of the judging team from quite a few years now, what do you think the key aspects you look at while you select a project, sir?
The first thing I look out is the passion with which the child is speaking. From that, I understand whether it was the child's own idea or it was a borrowed idea. Right? Borrowed in the sense, even if they have got a clue as to what it is, it doesn't matter. But if the project itself has been made by somebody and the child is vocally speaking about it, then we are able to easily distinguish. That's the first criteria. The second is whether that concept is really innovative. If it is really innovative, and if the child has not been able to perform to the level what is desirable, still we as a team usually see to it that if it has a potential to represent Team India, we try to select that project and try to mentor that child in such a way that the Team India is recognized at the international level. So these are the two very important things. And we look at the methodology. Methodology should be good, quite distinct, with suitable replicates, and no thermocol models and no plastic things to try to tell the repeated things. Like the repeated things which come is I gave this feed to the animal grew up this big in size. Now this has become too repetitive. Children, you must understand. A good question you asked, Shrey, because when we are evaluating, we are not just evaluating animal sciences. We are evaluating about 21 categories. And in the 21 categories, there are 100 students. We will get about 1,000 applications. First, we are selecting from that 100. And from 100, there may be one or two from animal sciences or 5 animal sciences. There may be 20 from computer sciences, engineering, environmental sciences. But finally, we are looking at not animal sciences or computer sciences or plant sciences or any of those. We are looking at Team India. And Team India is 20 participants with potential projects, which probably will have a sort of a make a mark at the International Science and Engineering Fair, where Team India would make a good representation. So we have to be above subject when we go for evaluation. So children, whatever subject you are appearing, then you should make it very clear that you have a strong foundation and a strong project from where you should be able to command, yes, I have a place for the ISEF in Team India. Is that clear? That is what we look.
Right. Thank you so much, sir. I think that's a very good answer. I'm sure the students will certainly gain from this particular, because we get this particular question from students. They always wanted to know what different they can bring into their idea, right? Okay, any more questions, participants? Else, I think it's time we can close the session. One last question. Please raise your hand.
Aditya.
Aditya Dioskar. Please unmute yourself, Aditya, now.
Yes, sir. So, good evening and thank you for the insights provided. So, I just wanted to confirm that these webinars which are going on, Could
Yeah.
you please tell the approximate date on which it will be for computational biology and bioinformatics?
Okay, so as of now, I can see the calendar as per my calendar. For computational biology, it is not planned yet, so it's not happening at least till 11th. Till 11th, it is not planned. 11th of September. So it will be done maybe from the second week onwards. After 11th, you can look for the calendar. Okay. We'll be putting
And
this calendar on the website as well very soon.
thank you.
Yeah.
Thank you, Aditya.
Okay.
All the best.
Thank
We
you.
have a question from Mahesh. I work on some research project on milk yielding in animal. We can show only result, but can't showcase the project. If I got the question right.
Can you repeat the question again?
I work on some research project on milk yielding in milk animal.
Good.
We can show only results, but
Yes.
can't show the project.
Yeah,
Is it
you
okay?
can't. Yeah, I can understand. He can't bring a cow and show it to us. He can only show us.
Okay, yeah, as a physical project.
But
Yeah,
you can
that is
always have a
completely fine.
video and you can have the pictures and you can have the data. But that it should be related to something. You
Right.
can't say that this breed gives this much of milk, this breed gives this much of milk.
Yeah.
It need not necessarily give, because based on genotypes and based on the climatic regions, based on the feed, there's a lot of variations which can take place. So if it is related to some real good factor, and many people work with the A1 quality of the milk and the A2 quality of the milk. But rather than that, if you can develop a sort of a simple mechanism of something like a test strip, which can identify the quantity of A2 milk in the milk which is being supplied, that could be a great contribution to people, because people now want to have milk with more amount of A2 rather than A1. So if you can work on that aspect, that would be great, and that could be a wonderful test which can be developed.
Right. So I would like to add on it, sir. Mahesh, you can always use the video. Okay, though you can create a video out of it, because in if you have not submitted your project yet, once you will go on our website and submit the project, it will ask you to submit a 90-second video, okay, of your project. So while you create a video, probably you can share, add the video part, whatever the work you're doing, or if you have actually devised something, you can show that product or show how the milking is happening, right? And along with the other information which you would like to share in the form of presentation, that you can do. So it's okay. So he's also mentioning we work on nutrition supplement to milk animal. Is there?
Yeah, milk animals, the animals which milk. Yeah,
Okay.
nutrition supplement is a very regular and normal topic.
Hmm.
Like it's not very specific, because conventionally people have stall feeding as well as free feeding. Free feeding is where the animal goes, grazes and come. Stall feeding, they're giving different combinations. So you will come out with a new combination. Maybe it's more very simple approach of a topic and may not have a much impact on any of your findings, unless you go into something else associated with it.
I hope, Mahesh, this has answered your question. If you still have a doubt, you can raise hand. Any other participant have a question? All right, thank you, Mahesh. Okay, so with this, we would like to pause the session here. And really, thank you so much, sir, for such informative session. And we hope that we'll keep conducting these sessions to benefit students on various information on various subject areas. And we look forward to have another session with Smile, sir, maybe sooner on a different topic. Uh, Thank you so much, participants, for joining us today. We hope that you follow us on our social media channels and you will get to know about our multiple webinars. Please join them and look forward to seeing you again. Thank you. Thank you so much.
Thank you.
Thank you so much, sir.
Thanks. Bye.
Bye, sir.
Thank you, Shaurya. Thank you.